Hi everyone, my name is Zachary Wakefield, and today I'll be presenting my project on how informing cancer prognosis prediction models using alternative RNA processing information can boost accuracy, and a bunch of other analyses related. So eukaryotic RNA processing is primarily composed of three main steps: capping, splicing, and the adding of a poly A tail. And so we're really focused on the splicing and start and stop aspect of this. And we focus on these kind of sets of alternative RNA processing events covering terminal exon processing and internal exon processing. And all of these events build an immense proteomic diversity and impact phenotype, often independent of overall gene expression. So to quickly cover what we use to actually quantify these phenomenon, first, the metric that we use is called a size score, or a percent spliced in score. And this is really simply just a relative usage metric. We see that, for example, in this here, the AFE1 is used in 3 transcripts out of 4, and so we give it a size score of 0.75. It's really basic relative usage metric. So the tools that we use are RMATs and the hit index to quantify alternative RNA processing events. And these use a combination of relative junction reads and exon covering reads. So RMATs quantifies the internal splicing events such as skipped exons, mutually exclusive exons, intron retention, alternative 3 and 5' splice sites. And the hit index quantifies and classifies alternative first and last exons. It also has a metric by the same name, the hit index, that is used to classify exon type usage. So whether an exon is used as a first exon, an internal exon, or last exon. And we also use this as a kind of alternative RNA processing metric here. So, as is known already, I just want to show that pan-cancer status is heavily associated with changed alternative RNA processing profiles and changed gene expression profiles. So here I'm just showing that there are signatures across control and cancer types that have large changes in what their max and minimum scores are. And here I just use max and min because I'm showing a mix of different ranges such as psi values, which are from 0 to 1, hit index values, which are from -1 to 1, and gene expression values, which are counts. We also know that many signatures are associated with a changed survival probability. So here we see that HNF1B and GREB1, skipped exons in each of these genes, are significantly associated with changing survival probability, dependent on their relative use. So to get to the question that we're really focused on answering is whether can we predict prognosis using alternative RNA processing signatures. So to do this, we looked at the Cancer Genome Atlas samples spanning 26 tumor types and around 9,000 samples. And on a cancer type and modality basis, so 243 models, we trained and tested MLPs with a Cox head. And so the input to each of these, dependent on the model, would be a size score or gene expression counts or the hit index. And along with each of these scores, we also feed in clinical information such as age, sex, race, and stage. And touching on some pre-processing, we remove minimal variance exons and genes. We min-max the log2 gene counts and min-max age. We one-hot encode the clinical data, and then we perform an extensive hyperparameter tuning. And after that, we train and test the final model, and we perform SHAP feature extraction. So delving right into the results, one thing really jumps out at us. And so this plot is showing the distribution of concordance indices, which is kind of a relative ranking of churn time or survival. And concordance index of 1 or C index of 1 is perfect, and 0.5 is completely random. And so one thing jumps out at us is that we see a lot of variance in terms of what is best performing. And the best performer is labeled on top of each of the box plots. And so we see a lot of gene expression. However, we also see a lot of alternative RNA processing, which is pretty interesting, suggesting that dependent on the cancer type, different information sources give different information. So Looking quickly at the SHAP results, we extracted out the top 5 most influential features from each of the models, and we're looking at the most common ones. And so we see a lot of already known therapeutic targets and cancer genes involved in this set, such as HLA-G. We also see some unknowns, which is pretty cool because it suggests this could be a new way to identify early potential therapeutic targets. And really hitting again, we see looking at the distribution of per cancer rank, meaning we ranked for every cancer which modality was best performing and which was least performing, 1 through 9. And so we see that gene expression is alone outperforming other information sources. However, a lot of other modalities are performing well. So to check that these models are actually using cancer-related information and pathways and genes, first we collected a set of 10 known cancer genes and searched for them in the top 150 most influential models from each of the 150 genes from each of the models, and we found in a lot of the cases that these are present. We also performed some gene set enrichment across cancer hallmark set and gene ontology biological processes, and we see a lot of classic cancer sets or cancer pathways such as EMT, KRAS, DNA damage and repair. So confirming that these models are actually basing their information on cancer-related biology. So one interesting aspect is that we see that many influential genes are not correlated across modality. However, we see that terminal exons show the highest association with each other, and that's here with the alternative first exons and alternative last exons showing the highest number when there are more than 2 modalities represented. So, given that we've built all these individual information models, we wanted to build multimodal models for each of the cancer types. So here we combine all of the different alternative RNA processing events and gene expression to build in one model. And so to do this, we developed autoencoders to kind of extract out latent representations of each of the different information paths to feed into an MLTP with a Cox head. And so we pre-tune all of the alternative RNA processing and gene expression autoencoders. We don't pre-tune the clinical data autoencoder. And all the autoencoders other than clinical data are frozen for the first 20 epochs and then unfrozen with a 1/100th learning rate of the Cox head. And we perform extensive hyperparameter tuning on both the encoders and the MLP. So the results from this show a few different things. The first is that looking at the gene regulation data or performance feature importance, we see a similar result to the single mode in that it's a mix, it's a medley. We see that, for example, in breast cancer, terminal exons perform the highest out of all the information types. However, this differs across cancer type. One thing that's been consistent is we see that clinical information is the most influential, with the top shared influential features across cancers consistently being race, gender, age, and stage. And I think this makes sense because these are associated with gene expression regulation pathways. So the last set of analyses I want to present are that ensemble models outperform the median performance from single modality models, where we see that across each of these, there's a pretty sizable increase in terms of the test concordance index when you're looking at the multimodal model versus the single modal model. And these are 2 different visualizations of kind of the same thing. So touching on some conclusions, the best predictor of prognosis is heterogeneous with regard to modality across cancer type. Models are basing predictions on biologically relevant information. And then also that multimodal approaches boost the concordance index and model performance. And a couple really broad in-scope future plans. The first is to identify potential novel therapeutics and then experimentally validate these. And then also using this multimodal approach, extend the current model to be a best-in-class prognosis prediction model. And with that, I want to thank you. Yeah, thanks.